Hi guys, I thought we would talk about the Regeneron monoclonal antibody cocktail because this is the one that's probably the most likely to for that people have access to. If you remember correctly, this is the combination that President Trump was given when he had COVID, and that was kind of early on in development under compassionate use. It recently got an emergency use authorization from the FDA. And this is a combination of two monoclonal antibodies, casirivimab and imdevimab. And so monoclonal antibodies usually, is just one clone, but they got two of them, so it kind of recapitulates a somewhat polyclonal response. So they have two monoclonal antibodies that attach at two different sites on the spike protein and neutralize in two different ways. And the idea behind that is that um, because of there's two sites it's recognizing, there's less mutational escape of the virus from a cocktail versus just one. If you have one, you could just have one mutation and escape. Okay, so this is the paper where they describe how they developed these monoclonal antibodies from Regeneron medicine. This is a paper with like 50 authors, which means a ton of people worked on it. Um, in order to understand this paper, it might be good for you to watch the monoclonal antibody video that I did a few days ago, because that will tell you what they're talking about in this paper. So in this work, we describe a high throughput effort using both mice and humans to generate antibodies against the spike protein of SARS-CoV-2. The ability to derive antibodies using genetically humanized VI mice. This is veloctamine mice. These are transgenic mice with a genetically modified humanized immune system. They make human antibodies even though they're mice, as well as B cells derived from convalescent patients enabled to isolate a very large collection of fully human antibodies with diverse sequences, binding properties, and antiviral activities. So they use two methods. They use the transgenic humanized mice as well as single B cell cloning from patients that had COVID-19 before. So they're using two different methods of generating human monoclonal antibodies. So they were generated with the following two methods. First, VI mice were immunized with a DNA plasmid that expresses SARS-CoV-2 spike protein. So basically, they immunize the mice. The DNA plasmid, that's essentially like getting the Johnson and Johnson vaccine. The Johnson and Johnson vaccine is a DNA vaccine. They essentially got a DNA vaccine, the mice, and then they were boosted with a recombinant protein composed of the receptor binding domain of the SARS-CoV-2 protein. So then they got boosted with a protein. That's kind of like the Novavax vaccine. So you can think of it, they got these mice, they gave them a DNA vaccine, and then they gave them a protein vaccine. Second, antibodies were isolated from peripheral blood mononuclear cells, so they drew the blood from human donors that had been previously infected with SARS-CoV-2. The VI mice elicited a robust immune response against the spike protein after immunization, so the vaccines worked on the mice as well. And so basically, the the human, the ones from the human donors, they were isolated. They extracted the white blood cells out of the blood, and then receptor binding specific B cells were fluorescent activated single cell sorted. So they use flow cytometry to sort them using a receptor binding domain fluorescent tag. All right. So then they isolated. They talk about, okay, yeah. So more than 200 of the VI. Okay, so screening yielded over 200 neutralizing monoclonal antibodies. So after they screened all these, they transfected them into Chinese hamster ovary cells. These are their production cell lines, and thousands of antibodies were isolated. So this is a ton of work. Thousands of antibodies were isolated, then screened for binding affinity and to see if they were able to neutralize a pseudovirus. So it's basically they engineer a virus that is not the full virus, but can infect cells. And this is a vesicular stomatitis virus that uses spike proteins of the SARS-CoV-2. So this is it's kind of a synthetic virus that simulates SARS-CoV-2 viruses. And they wanted to see which of these thousands of antibodies will neutralize them. So they yielded about 200 neutralizing antibodies. Then they sequenced all of them. These are the antibodies they got from the mice. These are the antibodies they got from people. And basically, they just gave them random numbers, and they were just checking for how well they neutralized. And it looks like, this one was really good. This one was pretty good, and this one was pretty good. These three were pretty good. Okay, so I'll get to the thing. Basically, the two that they ended up using was the 933 and the 987. This one they ended up calling casirivimab, and this one they ended up calling imdevimab. And these are the two that they went with in their cocktail. And from this, it's unclear why they picked those two. They were two of the better ones based on titers and neutralizing ability, but the best one was this one, and they didn't use that one. There was probably something about this they didn't like, the 989 one. Okay, so this is the fact sheet from the FDA of when they approved it, and this is the data based on which that they approved it for use. So they had a phase 3 trial with about 4,500 subjects, so big trial. And that's the advantage of having trials that are paid for by a pharmaceutical company. These trials are extremely expensive, but the pharmaceutical companies have the money to run these big trials. And big trials give you fairly clear answers. And so what they did is, if you look at this, they had two dosages, 600 milligrams and 1200 milligrams. There's a placebo arm. So in the 600 milligram trial, there was 700 in the treatment group and 700 in the placebo group, and about 1300 and 1300 in this group. And basically, the number of the percentage of subjects with at least one COVID event, which means either hospitalization or death, was 1% versus 3.2% in the trial. In the placebo, and then in the higher dosage group, it was 1.3 in the percent in the treatment group and 4.6% in the placebo group, which is about a 70% relative risk reduction. So if you get this early on, you can reduce your risk of hospitalization or death by 70%. So that is the basis on which they approved this treatment. This treatment is free and it's outpatient. America just recently bought like 1.4 million doses of this stuff. Regeneron must be making a killing on this. Um, they're making a lot of money because the government is buying it from them and then giving it out to people for free, sort of like the vaccines. And so what the indication is for is they want you to administer to patients 12 years of age or older with positive results that are high risk of progression to severe COVID-19. And this is their criteria for identifying high risk individuals. So if you get COVID-19 and you fit into this category, you can get Regeneron. And according to their data, it is likely to reduce your risk of hospitalization or um, death by 70%, which is a big number. The criteria are age over 65, obesity, uh, pregnancy, chronic kidney disease, diabetes, immunosuppressed, cardiovascular disease, chronic lung disease, sickle cell anemia, neurodevelopmental disorders, or having a medical related technological dependence. So I think like being vent dependent, having a tracheostomy, having a G tube, those kinds of things. This includes a lot of people, um, especially the obesity. There's a lot of obese people in America. And diabetes, there are a lot of people with diabetes. And cardiovascular disease, there's a lot of people with cardiovascular disease. So is quite a few people. And so, and they're giving this in mild and moderate disease. And most people with mild or moderate disease are going to do pretty well. So this is a and pretty expensive and technologically um, like intensive treatment regimen to give for mild to moderate disease. But that's the decision that the FDA has made. The other thing that's kind of new is the recovery trial, which is this large adaptive trial that's being run out of the UK. They did this in hospitalized patients, and they found out basically that in people that are immunosuppressed or do not mount a response that are hospitalized, it does help a little bit. Um, I think let's see, it's like 80%. Yeah. Uh, Rate ratio, so relative risk was zero point 0.8, so 20% risk reduction in people that did not mount a response. Overall, there was no difference. If you take all comers, there was no difference in death. It was 20% versus 21% in amongst these hospitalized patients. Um, and so, yeah, that didn't make a difference. But in the people that don't mount a response, it did make a small difference. So that may become a future indication if the FDA looks at that. So I hope that was helpful for people that are wondering about the Regeneron cocktail. Um, Now we know how it was developed, and what the indications are, and what the results of their clinical trials that gave them the emergency use authorization is. All right, thanks for watching.